Welcome to the first Water Talk of the new year, organized by the Water Institute at the University in Waterloo. My name is Roy Brouwer. I'm the Executive Director of the Water Institute. And before I introduce our speaker for this Water Talk, I would like to start by acknowledging that we're participating today from the traditional territories of the First People across the country. Here in the Waterloo region, I'm on the Haldeman Tract, land that was promised to the Haudenosaunee of the Six Nations of the Grand River. And here you see a map of the land. The Haldeman Tract is within the traditional territory of the neutral Anishinaabe and Haudenosaunee peoples. And as you can see from the map, there is a discrepancy between what was promised and where the original inhabitants of this area live and what was given as part of the agreement. The reserve is on approximately 19,000 hectares, whereas the original size of the land was almost 400,000 hectares. I encourage you to take a moment to recognize the traditional land where you are. Before I introduce the speaker today, just a few housekeeping items. Please add your questions to the Q&A box, not the chat box, and you can do this anytime during the talk. And then at the end of the Water Talk, my colleague Nancy Goucher and myself, we will get back to you and your questions, and the speaker will get to them. Just use the chat box for general comments and technical issues. The talk will be recorded and will be placed on our YouTube channel afterwards. So now it's my pleasure to introduce the speaker for today, Professor Amy Pruden. Dr. Pruden is Distinguished University Professor in the Department of Civil and Environmental Engineering at Virginia Tech. She has a bachelor's degree in biology and a PhD degree in environmental sciences from the University of Cincinnati. Her research focuses on converging microbial ecology and environmental engineering in identifying strategies to mitigate the spread of pathogens and antibiotic resistance in the environment. Her team's recent projects include creating an antibiotic resistance surveillance approach to apply DNA sequencing techniques to detect the spread of disease in watersheds impacted by storms, and to examine Legionella strains and how they operate before, during, and after outbreaks in towns and cities across the US. Dr. Pruden, her work on bioremediation, pathogens, and antibiotic resistance has been published in more than 100 peer-reviewed scientific articles, and she has been receiving awards for her work from different places. She's the recipient of the Presidential Early Career Award in Science and Engineering and the Paul Bush Award for Innovation in Water Research. And very recently, last year in April, she received the prestigious Recipharm's International Environmental Award. Recipharm is a very big pharmaceutical company. Professor Pruden, we're very honored and very happy to have you here with us today. I'm happy to hand over the virtual floor to you.
Well, thank you so much for the kind introduction, and it's really my honor to be here this morning as your invited speaker. I can't say that I mind the virtual format given all the winter weather we're experiencing. So yeah, I'm happy to share a little bit about some of the research we have going on that kind of converges what I'm calling a one water challenge with advances in Omics type approaches to understand microbial and public health threats. Here in my first slide, I have a little picture of one of our water systems that we have in the lab, and just give some appreciation for the scale of our research. We really, being in an engineering department, we want to design our experiments so that they are as translatable as possible to the real world and practical solutions. And so here we have some water heaters that you might recognize from your local hardware store. And so we have a pilot scale system I'll talk about later where we're looking at the colonization of opportunistic pathogens in water systems. So I guess a little bit of a downer to start my presentation off. So our U.S. Safe Drinking Water Act does not provide protection from opportunistic pathogens. This was the main conclusion of a recent National Academies report. So the water utilities, they're required to be responsive to this Safe Drinking Water Act, but there's some limitations because it only covers water up to the property line. And we'll see that there's all sorts of interesting microbiology going on beyond that property line that's important for public health. And also the Safe Drinking Water Act focuses on fecal indicator bacteria like E. coli and not on actual pathogens. And so I'll talk a bit about one particular portion of the distribution system today that's called the premise plumbing, the plumbing that's on the premises beyond the property line. And here is where the microbiology gets quite interesting, because you have a high surface area to volume ratio of the pipes, you have long stagnation times while the water's sitting and not being used if you're at work and going about your business, the temperatures are warmer, and you have poor persistence of the disinfectant residual provided by water utilities. And so it can really be ideal conditions for opportunistic pathogen growth. What do I mean by opportunistic pathogens? Well, the poster child is this Legionella pneumophila, and this causes a severe form of pneumonia that's deadly in about 10% of cases. And it's unique because it's not necessarily spread so much by drinking the water, but by inhaling aerosols. So you have to be concerned about showers and whatnot. And again, that's not something covered by the Safe Drinking Water Act, right? Similar for these other pathogens, Pseudomonas, Acanthamoeba, certain mycobacteria. These are also organisms that grow in the plumbing and can be spread by inhaling aerosols or skin contact and non-traditional routes. Over to the left here, I have a water heater, and this is a little bit different perspective on a water heater. This is a microbial ecologist's perspective. So we might think, oh, drinking water is sterile, definitely not sterile, low in microbes. Well, actually, there's quite a few. There can be on average about 10,000 cells per mil. And in a water heater, it's not always hot enough to really keep microbes at bay, especially with pressure to lower temperatures for energy savings. And because there are nooks and crannies and sediments, you have hydrogen that gets produced by these sacrificial anodes that are meant to protect the system from corrosion. Hydrogen's a good electron donor. So all sorts of opportunities for microbes to get a foothold in these water heaters. So the other big challenge that I've devoted my career to is antibiotic resistance, and really looking at it from an environmental perspective as a contaminant. So this is another huge public health threat, I'm sure you're all aware. And again, a microbial ecological problem in that the more we use antibiotics, antimicrobials, and we're pumping them out into the environment, the more we're putting selection pressure on microbes to develop resistance. And the challenge here is that, you know, at the rate we're going, we're not going to be able to rely on antibiotics as life-saving drugs in the future. And this O'Neill report is particularly grim, estimating that deaths due to antimicrobial resistance will surpass those due to cancer by 2050. And so the World Health Organization in particular has really urged that we need coordinated action. And I would make the case that a big part of the action that's needed is from the environmental perspective, and specifically a water perspective. So folks talk about One Health. That means looking holistically at human and animal and environmental health. There's parallels here with One Water. Now, in One Health, the piece that's really been lagging is the environmental piece, and we're trying to change that. And this UN Environment report came out in 2017 and really brought this to the forelight, and especially highlighted water environments and water as a thread by which antimicrobial resistance can thrive and spread. And so basically, mass balance, if you use antimicrobials in people and livestock, most parts of the world, most is going to livestock, it's going to go somewhere. It's going to go in the animal manure or wastewater treatment plants, and it's going to end up in the water. And so it's not just the antimicrobials themselves, but the resistant bacteria and the genetic material that they carry, including antibiotic resistance genes and mobile genetic elements that mobilize them across bacterial species. And of course, water is important because we depend on it for life, right? So there's plenty of opportunity for exposure via recreation and as a drinking water source. We've been especially interested in recycled water or water reuse because you've got a little bit of potential for short circuiting of any antimicrobial resistant bacteria and the genetic material in the sewage to carry over into the treated water. And so this is the work of Emily Garner, who did her PhD here at Virginia Tech. So, you've got, bluntly speaking, antibiotic resistance genes, antibiotics, resistant bacteria, they're in the sewage. And so even with advanced treatment, we don't know that it's really diminishing antimicrobial resistance. And we like to take this a step further and look at the distribution system. So a lot of times the distribution systems, especially for non-potable reuse, are purple pipes. So that's why I have a purple pipe here, that this can be an ecosystem with biofilms and robust microbial ecology where antibiotic resistant bacteria can thrive and exchange genes. And again, even though for non-potable reuse, we might say, okay, well, we're not drinking the water, it doesn't matter. Again, there are other exposure routes of concern. So inhaling aerosols and skin contact, and that you could be colonized and later experience an infection of sorts. And this is zooming in on a pipe biofilm ecosystem. So there are several studies out there that suggest this is really a key environment for gene exchange. So transduction, conjugation, natural transformation for ARGs to proliferate in microbial communities. So again, as engineers, we're hoping for some practical solutions. And so a big question that we're aiming towards is looking at whether water chemistry and plumbing conditions can be manipulated to desirably shape the microbiome of water systems. And what do I mean by that? So in a manner that excludes opportunistic pathogens, resistant bacteria, and ARGs. And so I was hoping to share with you some highlights of 4 different studies that give some perspective on this approach. And before I do that, I want to talk a little bit about metagenomic sequencing. So this is derived from next generation sequencing, which really caught on around 2010 or so. So, we've got a decade of data in this area, and it's really exciting. As a game changer that you can directly extract DNA from these environments of interest and sequence them and see what's there without having to pick primers or targets a priori. So advantages are, you can get the full complement of taxonomic and functional genes of interest. So that's really transformative. In terms of the research we're doing, we're really interested in markers of pathogen virulence, antibiotic resistance, and mobility of that antibiotic resistance. Now, that's not to say there aren't a lot of disadvantages. And you see that a lot in the literature, that people were excited to use this technique, but then get data and then maybe don't really know what it means. So there's some certain expertise required. There's some standards that need to be developed. There's limitations of databases. The data is semi-quantitative. And really a fundamental challenge is that the non-target DNA can really overwhelm the signal of interest. So One Water, thinking about conventional potable supplies, right? You take it from a source and treat it and deliver it, right? With potable reuse, which is really getting some traction, especially in parts of the world that are water stressed and, you know, realizing that the conventional approach is not sustainable, right? You can take wastewater treatment plant effluent and put it through multiple barriers, usually involving membranes, maybe even reverse osmosis and advanced oxidation processes to really hit the water with all the weapons you have, right? And then in the end, that it should be suitable for potable use. But there's shades of gray. So non-potable reuse, you know, maybe you don't need to spend all that money and energy to get that pristine of a product water, right? So you can do something in between, maybe without a membrane. So there's this system we've been looking at using biological treatment like denitrification, and also granular activated carbon, or GAC. You can use that as a physical means to remove pollutants, but also when it's biologically active, it can be a robust environment for microbes, and they can play a really key role in polishing organic carbon, breaking down emerging contaminants. And so there's a lot of interest in this system. And notice this is labeled a potable reuse system. So it's potable without a membrane. And this is something that we're starting to do here in Virginia. So this first study I wanted to share with you is the PhD work of Matt Blair. And we teamed up with Charles Bott at Hampton Roads Sanitation District here in Virginia, because they have this really interesting membrane-free activated carbon-based approach to water reuse. And Matt was interested in comparing, like I said, these shades of gray from conventional potable water to non-potable, and then different types of potable reuse water, and comparing that with wastewater effluent at one extreme and then bottled water at the other, since we assume our bottled water is clean, right? And so he started off by doing 16S rRNA gene amplicon sequencing. So this is a housekeeping gene in bacteria, and it's a nice marker that can be sequenced to kind of get a profile of the kinds of bacteria colonizing your system. And in this two-dimensional principal coordinate analysis plot, it's a way to simplify the data. So each one of these dots is about 10,000 DNA sequences representing a microbial community of these different types of water. So we've got our potable, reclaimed, which is non-potable, reuse, which is potable. And so interesting thing here is we saw that there was indeed a signature depending on the type of intended water use application. So the conventional signatures were down here, and then we've got the reclaimed or wastewater effluent here, and then shades of gray depending on the level of treatment for reuse. Now, we are also curious in what comes out of these biofilters, and you can see them clustered here. So it's a very different microbial community than what ends up in the effluent of the corresponding system. But again, highlights, you know, there's this robust, distinct microbial community that can really be a workhorse for polishing water for water reuse purposes. Now that was freshly treated water. Here's the same analysis after that water is passed through a distribution system. Again, here's purple pipes for non-potable distribution. And here we see it's a bit more blurred. There are distinctions, but the microbiomes tend to converge a bit because I think distribution systems themselves represent a unique habitat for microbes. And then I don't expect you to interpret all this data to the right, but just to show that we've dug into the sequencing data to try to say, okay, well, which are the microbes that are distinguishing these systems? So these are some of the phyla that we've noticed that have jumped out in the freshly treated samples, especially fibrobacteria show up in the reclaimed water, and Tenericutes show up in the reclaimed non-potable water. And then this is at the genus level, again, showing some of the ones that stand out. So Archobacter definitely showed up as a key indicator of non-potable water. So study 2 then, we're gonna look closer at the distribution systems for non-potable reuse water. And this is the PhD dissertation of Joyce Ju, and Sudesh Nagosh was a postdoc that helped with this project. This is taking it into the lab where we had a little bit more control over the conditions. So these are PVC pipe distribution systems where we brought in wastewater treatment plant effluent on like every other day. This was an intense project. This went on for 3 years. So we treated that wastewater treatment plant effluent again with our biologically active carbon filtration. Seems like a promising technique. We did that for 3 of the conditions. And then we also looked at 3 disinfectant conditions: chlorine, chloramine, and no residual. And then these are the same conditions without the BAC filtration. And then the distribution system has a 5-day residence time. And so we focused on sampling the treated water, the reservoir, 0.0, so right as it enters the distribution system, and then 0.5, that's about 5-day residence time. And so some highlights of what we found here. So this is relative abundance of antibiotic resistance genes. And so you might recognize some antibiotic classes that maybe you or a loved one has had to rely on to cure an infection. So like quinoline resistance, glycopeptides, beta-lactams. So the metagenomic sequencing allows us to profile the total numbers of all of these genes encoding resistance to these classes. And so on the left is a relative abundance. So the relative numbers of ARGs across these conditions. And so we've got the raw water from the wastewater plant, the reservoir after we treated the water, and then the 0-day water age in the distribution system, and then 0.5. And then we also sampled sediment. So it's interesting, these systems were really plagued by biological material that's sedimented in the systems. Highlights. So across the bottom are the disinfectant conditions. U is unfiltered, F is filtered. So we see that there's an interactive effect. So if you don't first pre-treat the water with the biologically active carbon filter, but you chlorinate it, right? So there's a lot of stuff in the water, if you will, and then you chlorinate it and it's reacting with that stuff. We saw elevated ARGs when the system's chlorinated in the water, biofilm, and sediment. And then here we zoomed in on mobile antibiotic resistance genes. So we assembled the metagenomic data to see which genes were capable of moving to different hosts. And again, we saw if you don't first treat the water with biologically active carbon filtration, but you go ahead and chlorinate it, the reactions there seem to elevate mobile antibiotic resistance genes in the biofilm and sediment. And here we zoomed in on what we're calling clinically relevant ARGs. So these are ones you hear about in the headlines that, you know, people have contracted some infection that's virtually untreatable with antibiotics. So like vancomycin resistance, certain forms of quinoline resistance, and then extended spectrum beta-lactam resistance and carbapenem resistance. Now we had a slightly different story, unfortunately. It was, if it is BAC-filtered and then you chlorinate, we had a little bit higher of these genes. So just when we thought we figured it all out, make sure you BAC-filter and then chlorinate, then this sort of threw a wrench into that. There were some interesting microbial ecological patterns. I think that fit with what we know. So the opportunistic pathogens that were annotated in the metagenomic data, so Legionella and Mycobacterium, tended to co-occur in similar conditions, and same with the enteric and fecal pathogens. And that makes sense because we know that the fecal pathogens are adapted to the gut of warm-blooded animals, whereas opportunistic pathogens are unfortunately adapted to water systems, right? And that's part of what makes them so difficult to eradicate. Something else that raised a flag with chlorine. So we plotted pathogen gene markers in the water phase versus biofilm. And we saw that if you look at the red, that's the chlorine condition. There were more of those that tended to fall in the biofilm side of the line. So it suggests that when chlorine isn't the disinfectant, you're actually enriching pathogen gene markers in the biofilm where they're somewhat protected from the chlorine and kind of sequestered there. And then these World Health Organization priority pathogens. So these are a lot of ones that are in the watch list for antimicrobial resistance. They did tend to be 2 to 3 logs lower in the BAC-filtered plus disinfectant conditions. So that's the open circles here, and chlorine and chloramine are on the left. So at the end of the day, if I had to give someone an answer, right, I would recommend this biologically active carbon filtration and some form of disinfection as a safeguard, but probably chloramine rather than chlorine, because chlorine raised the flags about selecting for antibiotic resistance. In this third study now, we're moving back into the premise plumbing, right? So we're looking at that portion of the distribution system beyond the property line. And that's, you know, the final stage before us consumers get exposed. So that's important to think about. But it's complex, right? I mean, talk to any, plumber, anybody in the construction industry, it's all over the place. It's the Wild West. But we'd like to give some guidance on, okay, what kind of pipe materials best protect you? Do certain systems like hospitals, should we recommend boosting disinfectants? What kind of disinfectants, right? And so when you have such a multifactorial array of concerns, but you're still trying to be at real world scale, it's complicated. And so we've developed what we've called these completely mixed pipe reactors that you're seeing to the right. And so we are able to accommodate right now 54 different pipes, right? And so in this experiment, we did 3 different materials, copper, iron, and PVC, and then lots and lots of replicates. And then looking at 4 different disinfectants, chlorine, chloramine, copper, silver, and chlorine dioxide. And it's nice because these are real world materials. Again, you can go to the hardware store, pick them up. Lots of replication, and translates well to real world conditions. And this is a project in partnership with Mark Edwards here at Virginia Tech, who's really world expert in premise plumbing and the science of which, and Abe Cullum is a PhD student that we're co-advising. Now, we looked at what naturally colonized these pipes, and then Abe also inoculated Pseudomonas aeruginosa, because that's an organism that is often a scourge of hospitals because it colonizes the drinking water systems and is often multi-antibiotic resistant and very difficult to treat. Okay, so a few highlights of results. This is looking at just the total bacteria, not necessarily pathogens, by measuring the 16S rRNA gene by qPCR. And we've got the 3 pipe materials, PVC at the top, copper in the middle, iron at the bottom. And then the colors are the different disinfection conditions. So copper, silvers in brown, chlorine dioxides in red, monochloramine is in, oh gosh, it's covered up on my screen, in blue, and the yellow is the chlorine. Okay, so in phase 0, we're just acclimating. Phase 1, really low doses of disinfectants, because there's some interesting theories about that, low amount of disinfectant is not enough to kill the microbes, but to make them mad, right? And to select for forms that are antibiotic resistant. In phase 2, we incrementally increased the dose of disinfectant until it reached the maximum contaminant limit. And then phase 3, we thought it'd be interesting to just stop disinfection altogether and see what kind of rebound there is. And so we noticed that PVC with chlorine, that because the PVC, it's really more inert than the other pipe materials and not reacting with the disinfectant, you get a lot more effective disinfectant residual. The second best was chlorine dioxide. So you see that here, the red is chlorine dioxide, yellow is chlorine. So much lower total bacteria levels. And then as you move into copper, it's not as effective, and iron, it's barely effective at all, because iron's a really reactive pipe material. And so it was really hard to keep disinfectant in there. Then looking at how the Pseudomonas aeruginosa behaved, on the left, we're looking at the numbers and normalizing the disinfectant to the control with no disinfectant. And so we see that, yeah, there's some couple logs decrease in the PVC condition. Copper a little bit more, maybe because copper is an antimicrobial itself. And when the disinfectant or the water reacts with the copper, it's released into the water, and that can have an antimicrobial effect. And then iron, some effect, but both the PVC and the iron rebounded when the disinfectant was removed, but copper seemed to still have antimicrobial properties, again, probably because of the copper pipe itself. Now, on the right is relative abundance, so normalizing it to the total bacterial population measured by the 16S gene. And this is interesting because it's showing a little bit different story in that you're more effectively killing off the background. And so relatively speaking, Pseudomonas is surviving better. And so you can see that in the free chlorine condition in yellow here, especially in the PVC and the copper. In terms of metagenomic sequencing, and we searched for pathogen markers, again, saw some interesting trends. So this mycobacteria group of opportunistic pathogens are quite interesting. They're phylogenetically, they're gram-positive, but they don't actually stain gram-positive. And that's because they have a really unique cell envelope that's very waxy. And that also makes them not very susceptible to disinfectants or antibiotics. And so people that end up with non-tuberculous lung disease have to take these cocktails of antibiotics for for months to try to treat that. And similarly, there's a parallel universe in our distribution system. So the PVC and copper here, you see with monochloramine especially, more than 50% of the reads were classified as mycobacteria. And so you're really, with the disinfectant, you could create a condition where they're the ones that survive. I did want to acknowledge that, metagenomics is not great at getting to the strain level. In fact, you really can't do that reliably with the standard approach. So we've done a lot of pure culture work too. And this is just showing that we looked at different strains of Legionella. So there's a strain that we isolated in a study we're doing following up a big outbreak in Quincy, Illinois. And we're doing experiments with it and realized that that strain's really copper resistant compared to, we'd been doing experiments with this standard 130B strain, which was pretty wimpy and easily killed by the copper. And so there's a lot of nuance here that has to be untangled. And this is the work of Yang Song and his PhD. Getting back to the metagenomic analysis of the pipes, and this time looking at ARGs, we also saw some interesting patterns. So again, some disinfectant conditions really enriched ARGs, so PVC with chlorine dioxide, copper with free chlorine, iron with chlorine dioxide. But it's interesting, it was reversible. So remember, at the end of the experiment, when we removed the disinfectant, the ARGs went back down to the initial levels. And here's some fun pilot data we have. So, hopefully you're getting the sense here that it's not just about the pathogens, it's about the pathogens as part of the broader microbial community that they're members of. And so we're really quite interested in understanding, again, how we can select for a beneficial microbial community. Maybe you call it probiotics for plumbing, whatever you want to call it. But in this pilot experiment, what we did is we took the feed water to the CMPRs and we pasteurized it. That's the line in red, or just fed it unpasteurized. So there's a nice healthy microbiome from the BAC filter going into the pipes. And you can see that Legionella was universally lower in all the pipe systems where there was a robust microbiota entering the system. And in the final study, I want to talk about going back to water reuse. This is the PhD work of Ishi Keenum. We were looking at two water reuse systems, both. Well, the one is non-potable at the top. That's a system in Florida. And the one on the bottom is potable, but not using a membrane. So both use some biological treatment. And so we're interested not just in the treated water, but in the system on the top. In Florida, they distribute it to homes and businesses for irrigation and other purposes. And then the system in the bottom, we're recharging the groundwater here in Virginia to deal with some aquifer issues. And so we did really comprehensive profiling throughout the treatment, distribution, groundwater injection. We did metagenomic sequencing. We also wanted to do some culturing and focusing on what are called these escape pathogens, because they're of interest for antibiotic resistance. And then Kong Sia is a collaborator of ours who is an expert in environmental chemistry, and she's done some non-target screening of pharmaceuticals for us. So a little overview of the data. So in Virginia, in the system here, again, this is a potable reuse system with BAC filtration. We cultured for these fecal pathogens, so Klebsiella pneumoniae, Enterococcus in the middle, E. coli to the right, with and without antibiotics, so that those are shown in red. And the nice thing is that we've seen generally a decrease through treatment. So that means, wastewater treatment and the added measures for recycled water treatment are reducing these targets. And we also see that the resistant forms they're about a log or so lower than the forms that grow in the plates without antibiotics. And these are just two different sampling events in the top and bottom. And in Florida, we similarly saw reduction through treatment, but we saw what looked to be some sort of a rebound in the distribution system. So again, highlights, that that's a robust environment for microbes to grow. So same for E. coli. We were also interested in trying to culture what we called environmental pathogens. So really the kinds of opportunistic ones we've been talking about. And these are ones, Pseudomonas and Acinetobacter in particular can have niches in water environments. So we're interested if that might give them more opportunity to acquire antimicrobial resistance. That's something we're looking at. But really what we noticed is that it was hard to culture these organisms from these water systems. I mean, this is a big endeavor doing culturing because, you've got to process the samples within 48 hours. And this is a lot of samples. And then the methods just aren't very optimized for water and wastewater, right? So E. coli, Enterococcus, right? We've got standard methods, but these ones, the methods are really better geared for clinical samples. So I think some work needs to be done on the culture methods. And so this is in the Florida system. And again, we saw some evidence of regrowth of Acinetobacter, but it was just hard to detect these organisms and have statistically supported conclusions. From a metagenomic standpoint, and we're looking at antibiotic resistance genes again, on the top is relative abundance. And in the Virginia system, we saw a nice removal of ARGs, some evidence that ozone, which is a big stress on microbes, right, that might be selectively enriching antibiotic resistance. And then also chlorination came up as a flag again that could be selectively enriching antibiotic resistance. On the bottom is what we're calling absolute abundance. So we've back calculated ARG copies per milliliter. And this is, when we're working with people in the water community, they prefer these units because it's more intuitive. And as far as, if we're going to move towards risk modeling, it's more amenable for that. But even then we saw a little bit of uptick with ozonation, and we saw that levels were pretty low in the groundwater. But in the Florida system, again, not only evidence that the pathogens could regrow in the distribution system, but both relative and absolute abundance showed that antibiotic resistance genes could be being enriched. So some closing thoughts for you all. This is a grand challenge, and I think things are really starting to come together to rethink water quality, right? Not all microbes are bad. I think historically in the water industry, it was like the only good microbe is a dead microbe, right? Well, that's not true. We're seeing, first of all, you can never have sterile water, right? And once you move beyond that, you can think about, well, who are the good guys, and how can we design water treatment, disinfection, and pipe material to desirably shape microbiomes? And so, how can we do this more intentionally? Can we think of it in a probiotic sense? Is the world ready for that? And with next generation DNA sequencing, I think there's a real opportunity here. Yes, we've relied on culturing fecal indicators, E. coli, Enterococci, very useful information, but it can only take us so far. And so I think there's a lot of potential with next generation DNA sequencing, machine learning, to enhance our water quality monitoring capabilities. Now there's limitations here we're working on too, and I'm happy to talk about those in the Q&A session. Just let you know, we have some collaboration with Li-Ching Zhang and Lenny Heath here in computer science. And we're particularly interested in looking at metadata and really kind of a big data approach to understand across these systems, you know, what are the key drivers of microbiomes. And just wanted to bring to your attention this National Academies report on Legionella. And we did have a couple Canadian participants in helping put this report together. But I think it really does well to highlight that we need a systems perspective in thinking about our water quality. So, you know, not just disinfectants, but thinking about plumbing materials, hydraulic designs, temperatures, and also means that people can be exposed, like by aerosols, and, you know, thinking beyond just ingesting the water. And we've got 3 projects funded by the Water Research Foundation that are looking at advancing the use of metagenomics and water quality monitoring. And we recently published this paper in Water Research that's a very, very comprehensive, dense summary of the state of the knowledge and approach to next generation sequencing approaches to evaluate water quality. So I hope that's a useful reference. And finally, in case there's any students out there looking for a good place to do an interdisciplinary PhD, we recently got this training grant from our National Science Foundation that I'm quite excited about. So it's a convergence approach. So that means not just interdisciplinary, transdisciplinary. So we're looking at data science, environmental science and engineering, policy, microbiology, right? So weaving these together, but also working with stakeholders. So farmers, wastewater treatment plant operators, NGOs, doctors, everyone else to kind of come together and to formulate robust science-based solutions to the problem of antimicrobial resistance. And so I have many folks to thank for this body of research that I've been able to share with you today, and especially thank funding from CDC, NSF, Water Research Foundation, and various institutes at Virginia Tech, including CZAP, NanoEarth, and ICTAS. And with that, I'm happy to answer any questions that you may have.
Thank you very much, Amy. Thank you for this fantastic presentation. It was excellent. Thanks for sharing your research with us, and also putting it into a broader context. I like the picture that you showed at the end also about the upscaling and the systems approach. We have some time for questions. There are 4 questions I see in the Q&A box. So I propose I take the first one. I will just read it out loud to you, Amy, and then you can take the question. And then the next one can be taken by Nancy. So just going to the first one is from Masha Masheki. I hope you pronounce your name correctly. Can we remove pollutants by biological catalysts, is this question.
Okay, so trying to make sure I understand the question correctly. So from my perspective, I think of microbes themselves as catalysts, right? They're doing stuff. They have enzymes, and so it's about getting inside the heads of the microbes, right? They're not working for us, but if we can figure out, you know, the conditions that make them happy and make us happy, right? So I think the biologically active carbon filtration is a great example of that. So they like to colonize the activated carbon, and that forms extensive biofilm and a high surface area of catalytically active microbes. And then if you put ozonation in front of the BAC filter, that basically chops up the food for them in little pieces, right? So, you know, the kinds of things we'd like them to remove, like these pharmaceutical compounds, they're big, complex, low concentration compounds. But if we can chop them up with ozone, we kind of find a nice harmony there. And yeah, so that's just one example of taking advantage of microbes as natural catalysts. But, you know, there's all sorts of examples with environmental remediation, oil spills. So yeah, no, I'm a big fan of microbes.
He's referring to E. coli for catalysis of polymers in the chat box. I don't know if that's still relevant.
Yeah, no, I'm not familiar with that. I tend to just think of E. coli as a fecal indicator, but of course it's also a workhorse in the lab and can be engineered. People play around with its genome all the time. So yeah, no, I'd be interested to learn more about that. I know people are interested in thinking of wastewater more as a resource or generating value added products like polymers. So yeah, no, I'd be interested to know more what you have in mind if you want to email me afterwards.
Thank you.
And
Nancy,
this next question, the next question is from Bruno Garcia Liva. What's your opinion about advanced oxidation process in biological pollutants?
Okay. Well, I think they're a vital part of a multi-barrier approach. And I think it's catching on more here in the US. I know in Europe, they're big fans of ozone in particular. Now you saw some flags in our data, like ozone tended to elevate some ARGs, and we don't know exactly how that translates into risk, but it raises a flag. And I think if it's part of a multi-barrier approach, right, we're combining it, we follow that up with the BAC filter and the ARGs go down and pathogens go down. So I think, you don't want to use them in isolation, but if it's applied strategically as part of a multi-barrier approach, I think they're really valuable.
There is another question from an anonymous attendee. The question is, how sustainable would chloramine treatment as a disinfectant be as opposed to chlorine following BAC filtration? Could we expect a similar trend of sequestering selection for AMR in the microbiome given enough time?
Okay, so chloramine versus chlorine. So just so folks are aware, chloramine is formed by mixing ammonia and chlorine. And that really caught on here in the US in the last 20 years or so, because it's a bit more persistent than chlorine. It's a little less reactive with the pipe materials, right? Like chlorine tends to get eaten up by iron right away. Chlorine persists a little better. It's thought to better penetrate biofilms. But there have been some drawbacks, like the nitrogen that you have to add, the ammonia. In warm conditions, it's really susceptible to nitrification. So ammonia is an electron donor for nitrifiers. And so what ends up happening is, even though in ideal conditions it's more persistent, now suddenly it's much less persistent because the nitrifiers just break it down. But I guess, so from what we were showing, like when we applied it in the premise plumbing as a booster in the pipe experiment. So again, this is an analogy, like at a hospital, somewhere where you have vulnerable patients, you might actually apply it at the building level, not just at the whole city distribution system level. There it's more controlled, and in nitrification might not necessarily be a problem. And so at least in that experiment we saw, it seemed like a happy medium in terms of not raising as many flags with selecting for antibiotic resistance. It seemed to give a good amount of pathogen control, but again, not for mycobacteria. And we don't know that the mycobacteria we saw are necessarily the pathogenic ones, but certainly is cause for concern. So I guess there's not an easy answer to that question. I think, again, it comes back to a systems perspective. you have to look at a case-by-case basis and look at the combination of the water chemistry at that locale. You know, how corrosive is it, for example? You know, what is the other nutrient content? What are your pipe materials, temperatures? And you kind of have to evaluate it that way to see, you know, what's the optimal disinfectant for what you're trying to achieve.
Thanks.
Thank you, Amy.
We try to sneak in one more question. Okay, so if Mycobacterium remains after everything else is killed because of its mostly mycolic acids, is this possibly a good news indicator that most microorganisms have been eliminated and that treatment was effective overall? Are the methods used together able to distinguish between persistence, growth, and naked DNA to help answer this type of question?
That is an excellent question, and I think would be a good one for some follow-up study, right? So in this study, we could look at big picture patterns with the DNA, and, you know, so that's a very good point. The metagenomics is capturing the DNA from both live and potentially dead organisms and their naked DNA. So we couldn't directly distinguish in this study, and that's part of why we do some culturing to complement that data. So I think it would be good to look in future studies, doing a bit more culture work with the mycobacteria. Now they're very, very difficult to culture. And we're fortunate here at Virginia Tech, we have Joe Falkenham, who's devoted his whole career to culturing mycobacteria and is really an expert at that. It takes weeks, and then to differentiate the pathogenic strains and non-pathogenic. So that's part of why we like to use metagenomics, sort of screen and be like, oh, here's a concern, let's focus on that for culture and send it to Joe's lab. We are doing a little bit with some metagenome assembled genomes. And so that's where, if we do deep enough sequencing and do it strategically, we can start to put together the genomes and tell better which are the pathogenic ones or not. Because I suspect that whoever asked that question is right, that not all these mycobacteria are bad, and maybe a lot of them are good. And so I think getting methods to better resolve that would be very helpful.
Perfect, thank you. So the question was from Professor Josh Neufeld in our Department of Biology, just so you know.
Very knowledgeable question, I could tell.
Thank you, Josh, for the question. I'm afraid we have to leave it at this. We're 2 minutes to the hour. Amy, thank you so much once again for your amazing presentation. Excellent, and we're very happy to have you here today. To the audience, I hope you enjoyed our first WaterTalk of the new year.